Good day, everyone. I'm going to continue here where I left off with genetics when I started this topic. And we are going to do our very first genetic cross. And I'm going to go into all the details as much as possible so that we're going to use this one genetic cross to help us solve issues that we face in many different types of genetic crosses. So this is going to be like the foundation kind of genetic cross. We'll start with this question that I have here. Our question says, in guinea pigs, dark fur color is dominant over white fur color. Do a genetic cross to determine the phenotypes of the F1 generation of a cross between a male guinea pig, heterozygous of fur color, and a female with white fur. Now, first things, we are going to approach this question as if we are doing it practically. So we are going to physically go and try to see these guinea pigs physically and see how you can cross them like you're doing a natural breeding with two guinea pigs. So my first question is, when you get to these guinea pigs, what do you first see? Do you first see their genes or you first see their physical appearance? I'm sure the first thing you notice will be their physical appearance. That is why in our genetic crosses, the first thing that we will write will be the phenotype, because practically that's the first thing that you're going to come across, going to see their phenotypes. Secondly, during the course of our genetic cross, you'll realize that these guinea pigs that we are going to cross will have to produce offspring, will have to give birth, theoretically, to offspring. So because they will produce offspring, they will become parents. For that reason, we shall write P, which stands for parents. And since our genetic cross says these guinea pigs will give birth to the first filial generation, the F1. We're looking for the phenotypes of the F1. If they have to give birth to the F1, then themselves they will have to be the first parents. We'll write it as P1. So our first line here should read P1 phenotypes. Nevertheless, I always say it's much better to work out the genotype so that we can always backtrack and determine the phenotype by using the genotype that we have. And this is a careful way of avoiding saying or writing a phenotype which does not correspond to the genotype. I'll explain this because many people run into this kind of mistake. And it's simply because they like working out the phenotype as they read before trying to work out the genotype. The phenotype and the genotype must speak the same language. You would understand that the phenotype is the physical expression of the allele combinations that are found in the genotype. Keeping in mind, here we're dealing with complete dominance. So one trait is completely dominant over the other. Now, let's look at our question. When we say our question, first ask yourself, in this question here, based on the two guinea pigs that were given to us, what was given? What information was given to us about these guinea pigs? Did they give to us the genotype? Or were we given the phenotype of these guinea pigs? Which one was actually given? Now, here again, most people just jump into the conclusion and say that they always give us genotypes and all of that. The genotype is just the allele combination. The genes, the nature in which the genes are combined in a particular individual. And from there, it will determine the phenotype, how physically this person will express what this person has as in terms of genes. That's just bluntly said so that any person can easily understand what it means. So I'm going to go through this so that we avoid certain things. Whenever we talk of phenotype, people tend to say, for example, I have something that looks black, and then they know somehow that that thing was talked of as being maybe heterozygous or homozygous, and then you will see people writing on phenotype heterozygous black on the phenotype, or heterozygous green, or homozygous blue. Now you cannot use the words hetero or homo when you are describing the phenotype, because when I see hetero means different. Homo means same or similar. So when I say same or similar, it means I'm comparing two things already, two or more things. But therefore, we can't talk about one phenotype for one individual and term it as hetero or homo. It's like you're comparing one thing with itself. It's not even possible. Secondly, I want us to look at this diagram here. Let's say I'm drawing someone who is tall. Okay, you'll find here. This is someone who is very tall, like that. Don't mind the drawing. Just by looking at this diagram here, is it possible to tell that this person is either heterozygous or homozygous tall just by looking at this diagram here? The answer is no. But then whenever we are writing out phenotypes, we find it very convenient to write things like homozygous or heterozygous. But if you look at this diagram, just on the diagram alone, you can't even tell whether this is heterozygous or homozygous. What am I trying to say? All I'm saying is that these words are not meant for phenotypes. Like I usually say, you cannot look at someone and say that the person is heterozygous. Yellow bone, for example. No. All you see is yellow bone. Those who know. So, hetero, homozygous, we cannot give it to the phenotype. But let's see something else. If I should write, for example, this, capital T, capital T, immediately I ask you, is this homozygous or heterozygous? You'll see it's very easy to say homozygous. Why? Because when I look at the two letters, they look the same. They look similar. Of course, it's easier to say hetero or homo here because I see first two items, and then it makes it very possible for me to compare them and term them as being same or similar. If I have capital T and small letter t, lowercase t, then I can easily say here that they don't look the same, they don't look similar. Then I can easily use the word hetero. Hetero because I'm comparing two things. I physically see two things that I can compare. And if I have lowercase t, I can easily say homo because I'm comparing two things. What are we saying? We're saying that these terms, hetero or homozygous, belong to the genotype. So we only use them when we're talking about the genotype. Now, coming back to our question, data site. The first question that I asked here was, what did they give us about these two guinea pigs? Did they give us the genotype or did they give us the phenotype? So with what we've learned just now, what we use to denote genotypes and what we use to denote phenotypes, when I look at my question here, I see they say do a genetic cross to determine the phenotypes of a male guinea pig heterozygous. It tells me that here, the moment I see this, and it means they're talking about the genotype. For fur color, and a female with white fur. Now, with this female here, I don't see anything like heterozygous or homozygous. I see straight they're talking about a color, white fur. Since there's no hetero, since there's no homo, I know clearly now that here they're talking about the phenotype. So, back to my genetic cross. For the male, they talked about the genotype. For the female, they talked about the phenotype only. Now, in our previous video, we saw how to decide on what letters to use. My word of interest here is dark, because they said it is the one which is dominant over white. So I asked myself, how do I spell dark? Dark is spelled starting with letter D. So wherever I want to represent the dominant allele, I'm going to use capital letter D. And wherever I'm going to want to represent the recessive allele, I'm going to use the same letter, but this time in lowercase d. So hetero here would be capital D and lowercase d. That will be for the male. And I go backwards now and ask myself, phenotypically, what will this produce phenotypically? So it will give me, because dark is dominant, then I'll have dark fur for the male. And when I see for the female here, we saw that for the female, they didn't give her genotype, they only gave her phenotype. So I'm going to cross dark fur with white fur, which was directly given for the female. Okay, so my phenotypes are set. Now, one thing that we need to correct here, usually we find people crossing like this, dark and white, or black and cross with white, as if they are fighting against racism. So if I am your teacher and I see you writing something like this, like I usually say, I'm going to write there for you, Black Maguinia, so that you can go home and show to your parents how your life is progressing. What we're trying to correct here, please, is this. Whenever you're answering questions, always answer as if the person reading your script doesn't know anything about what you're talking, so that you always put yourself in the posture of teaching and not answering directly. Because if you answer as if you are teaching, then trust me, you will not leave any stones unturned. But if you answer as if the other person knows what you're talking about, then you will never give all the full answers, because you assume that when you say black, I know exactly what you're talking about. But no, when I pick up your script, say on the floor somewhere, it fell off somewhere, and then I came across it, I should be able to know that you're talking about dark fur. I should be able to know that you're comparing dark with white fur, not just two colors. What exactly is it that is dark? That's what we're talking about. Now, the genotype for the female here is not given, but that's not the problem. The question has been carefully asked. If you realize, they say here, dark fur is dominant over white fur, which means white fur color is recessive. There is only one possibility for a recessive trait to be expressed in the phenotype. Only one possibility. The only possibility is that the alleles that control that recessive trait must be present in a homozygous state. You see two letter Ds, or at least the dominant allele should be absent. It means if I put this, the moment I add a dominant allele there, then the phenotype will be the dominant one, which in this case is the dark fur. So for me to get the white fur color, it's a must, I must have double recessive. So it's very convenient also to set a question in this way, because without stating the genotype, because we know that there's only one option for white fur. Let's say, for example, I had set a question which says a dark fur guinea pig was crossed with a homozygous white fur, and dark fur being dominant. It will be very ambiguous and it will lead to indecisiveness, because when I say dark fur, you would realize there are two options to dark fur. It could be double, capital letter D, or in a homozygous state, or it could be in a heterozygous state. And now we'll be set into asking ourselves, which genotype here should we consider? Because not both of them will lead to the same phenotypic expression. So that's why when we set questions, we make sure that with the dominant alleles or with the dominant phenotypes, we state clearly the state of the genotype. I cannot state by just saying a dark fur male was crossed with a white fur female. For the white fur female, there will be no problem because there's only one option, which is this one. But then for the white fur male, it will leave us with two questions. Is it the first one, or is it the second one? That will be the question. That's why we carefully made sure that the genotype was stated by saying heterozygous. So our genotype for the female will be this one here, because it's the only possibility, only thing that can produce white fur. Now, so here we're going to write two small letter D's. Now, the reason why I started working out the genotype, especially for the male, before working out the phenotype, is because here is where we usually make errors. You'll find someone that's written there for the female white fur, and here writes a genotype like this, and then the person moves on. You should understand, the moment you make this kind of mistake, if the question is out of 6 marks, the maximum here you will be able to get will be 3 marks, unless you perform some other miracles, because the genotype here is already wrong. The genotype is wrong. And by the way, like I said, the genotype and the phenotype must be speaking the same language. Here on this line, you said this was white fur. But down here, you're writing a genotype that cannot produce white fur. That's number one. It means that the language spoken between these two here is not the same. It can't be possible. Secondly, what you have also done is that the same genotype here, which is producing white fur, the same one here, same genotype on this side also miraculously is producing dark fur. So before you move on, you should ask yourself these questions: how is that even possible? So that you can check your work and realize that, no, here you made a mistake. To avoid such mistakes, do like I did with the male here. You start with the genotype if you were given, and then you work backwards, and then you write the phenotype. You work out the phenotype from it, because you will see that because of the dominant allele which is present in that heterozygous genotype, it will always produce a dominant phenotype, which in this case is dark fur. So to correct, we had here for the female, it will be 2 small letters D, 2 lowercase d, and then here we should not forget to put our X. Now, another mistake that we usually make, sometimes we'll write here the phenotype just because it's so long. So I'm going to write here dark fur male. And then you see it's very long, it's extended. And here, come here, and then we do our cross, and we write the genotype here. Now, that looks very wrong. You would understand there are 2 individuals here, and each one should have its personal phenotype and its personal genotype. You realize that you've given here 2 genotypes for just one individual. Doesn't make sense. Where is the genotype for this person here? Everything has been shifted to that side. So these are the kind of mistakes that we shouldn't make. So we need to correct that. Each individual, its own genotype. So I'm going to put here for the female. Question. Is meiosis part of reproduction? Answer is yes. And why is that so? It's because meiosis leads to the production of gametes that we also call sex cells. Specifically speaking about sexual reproduction here. So if I rephrase, meiosis part of sexual reproduction. Our answer will be yes. So we need to produce gametes through meiosis, through the process of gametogenesis, divided into two here, spermatogenesis, which produces sperm cells, and oogenesis, which produces ova. So those are our gametes. And therefore, when I look at genetics here, our genetic crosses are simply a summary of the process of reproduction, which is the process through which individuals produce new generations of themselves. Now, therefore, meiosis would have to take place here so that we can produce gametes. And during that process, alleles that are found in the genotypes would have to separate like that. And what exactly is happening is this. What's happening is, if we want to make it practical, we have chromosomes here. You should understand chromosomes are just, I could summarize it to being DNA. It's DNA. And DNA has sections that store information that we call genes. So the genes here, I've talked about alleles before. I'm taking the male here, for example. What we are having here, in that male received the dominant allele, say, from one parent. I'm representing here a pair of homologous chromosomes. The male received a dominant allele from one parent and received a recessive allele from the other parent here. So during meiosis, these chromosomes would have to separate. You should notice I've written just one letter in each chromosome representing a gene, but these chromosomes have close to 1,000 genes. And the letters that we are using here are only there to represent the genes that we are interested in this particular genetic cross. That doesn't mean that there's no other genes scattered all around the chromosomes. There's so many, but right now I'm only dealing with the one that gives us fur color. That's why I'm representing it. So during meiosis, these will separate and land in separate gametes that will form. What law is applying here? This is Mendel's first law, the law of segregation. The law of segregation, which states that when two parents reproduce, the factors of inheritance are segregated. The factors of inheritance that Mendel was talking about, where today we know them as genes. These factors or these genes separate and go into different gametes. So that's what will be happening here. Our alleles will be separating to end into different gametes. So I'm going to have it here and here. Those are the alleles separating. Now, corrections first. When we are doing our genetic cross, this is another thing I keep seeing, other mistakes that we keep doing. We need to correct them here. When we are drawing, somehow some people have the habit of saying they are using a template, which to me is just equal to cram work, which is the thing we must avoid the most. There's no such thing we should ever use or that we call template. A genetic cross should be developed step by step. And it's also because we like skipping steps that we get mixed up completely. Step by step. The template, usually you'll find people writing, they write phenotype, genotype, and then they come somewhere here and write meiosis. And when they come at this step here of producing gametes or forming gametes, this is what will happen. Draw here. And then here, then you have the gametes there. Now, when you look here, the gametes that you've drawn, they fall on the same line with meiosis. So you are labeling that thing meiosis instead of gametes, simply because as a result of cram work, you went on to draw already all the steps that will be required in your genetic cross, right? Fertilization, F1 genotype, F1 phenotype, and all of that. So the best thing that I always recommend here we should be doing is, before writing meiosis, we are going to separate here, going to draw that line of the arrows completely. And in the space, because there will always be a space here, I can easily squeeze in meiosis. Comes easy, and every fool can see that that is meiosis taking place. And whatever will form down here, I can easily write the gametes. It means everything aligns properly. Now, another thing. When we are drawing our lines, I know some of us, our lines are too straight. Then we draw also like that. Very straight lines, like that. To show to the world that we are the chosen ones of God. So you will find that you draw your gametes here and all of that. The question here is there any difference between this line and this line here? You see, everything just looks like genotypes. These are not gametes. Unless you were lucky enough to put maybe a comma somewhere there to say to the examiner that they are somehow separate. But I don't recommend this. We must see that there was separation. From that first law of Mendel, separation, there must be segregation. Let me put back the X here. There must be segregation. So that's why I always draw this way. I physically make sure that there is some kind of separation here. And I'm going to have my gametes. Everyone can see them clearly. Right in between the gametes and the genotype here, I'm going to squeeze in meiosis, and everyone can see it. And here, I can now label gametes. And it aligns properly. Now, the next thing we are going to do. I know we like drawing these lines and crossing this way and all of that. I personally don't recommend it. Okay, it's a good practice. But if you want to do it, each time you draw a line at once, write out the outcome. D, capital D or uppercase D, goes with lowercase d on the other side here. And then I finish with that line, I write out the outcome. This one, I also do that. Finish with it, I write out the outcome. Same here, I do. Finish, I write the outcome. Same, I do. Finish, I write the outcome. Now, the reason I'm not too much a fan of this is because you'll find people who draw lines like that. You know, when you're babalas, too much incumbency, you can't draw anything straight, and then things just start falling apart. It looks messy. Or you'll find people who take a ruler and draw very neat straight lines like that. Very neat lines. Mine are a bit cranky, but then they will draw very beautiful straight lines like this. And all of a sudden you start seeing miracles appearing downward. Where they come from, we don't know. How they appeared here, we don't care. How they got there, we cannot tell. So to avoid that, please, I think the best thing to do is to use another way of representing our genetic crosses. But before we get to that, I want us to clarify something here. In this line up here, this is when the chromosomes are still in their full double sets. So this line here represents 2n. And if you count the alleles that we have here, in each of them we have 2 alleles. And in this line here, you realize that at each point we have just one allele. One allele. Here we're having two alleles at each point. So the line down here represents when the chromosome numbers have been halved. It represents the n line. And I want us to therefore realize that we left from 2n to n. We ask ourselves, what kind of process does this? What kind of process brings about the halving of this 2n into n? Or rather, what kind of cell division causes that? That kind of cell division is what we call meiosis. This is just to justify to you that truly what's happening at this stage is meiosis, as we labeled here. I said at the beginning, we must be able to account for whatever thing we are writing here. We don't just write things because we have crammed a certain thing called a template. So we must make it clear that's meiosis that took place. Now, additionally, when I was talking about these lines that we draw and that don't look nice and all of that, usually when I talk about this and say, please rather use the other one that we know of, the Punnett square, people get scared and then they say, no, the question said, do a genetic cross. Do a genetic cross. And that's why they want to start crossing things as if they're the ones who put Jesus on the cross. No. This, we should understand, the one that we did earlier, is simply what we call a schematic representation of a genetic cross. And we also have a Punnett square representation of a genetic cross. So they're simply forms of representation. There's not one that says this one is the cross and the other one is not the cross. In either case, we are always doing crosses. It's either we do our cross in a schematic way or we do our cross using a Punnett square diagram. So all of them are crosses. Please don't get scared. It's the same thing that we're doing here. But I prefer a Punnett square because it is much more neat. And although my handwriting naturally is a very poor one, nevertheless, my Punnett square, whenever I write inside of it, whatever I do with it, it always looks very neat. Okay, so first, because this is a monohybrid cross, Monohybrid monohybrid because there's only one trait being taken into account here. And the trait we're looking at here is fur color. So it's a monohybrid cross. So because it's a monohybrid cross, our Punnett square would have to have 3 columns and 3 rows. We need to understand this. Remember these numbers. In this case, it's 3 by 3. If we are dealing with a dihybrid cross, it will be 5 columns, 5 rows. It will be 5 by 5. We need to remember this figure so that when we are faced with an exam, we don't waste time asking ourselves that, but how many columns were here, how many rows were here, and you find people drawing 1,000 of columns and they get everything just mixed up. So I'm going to simply draw here. I'm going to draw Punnett square. 1, 2, and 3. Then 1, 2, and 3. Those are my 3 columns, 3 rows. Here, if I want, it's optional. It's not optional anyway. You have to gametes in here to show to the whole world that the gametes that are had here are the same ones that are brought down here. The gametes that are had here at that other line are the same ones I'm going to insert in my Punnett square diagram. Now, next, I'm going to take the gametes as they are and put them inside the Punnett square. So I've taken these ones and I've added them here. I'm going to take these ones and then I'll put them here. Small d and small d. Or I can decide to do it otherwise. It doesn't matter. I can decide to take this one and put it here, and this one's here. So I can do it this way. Sorry, my eraser is messing things up. So I can do it the other way around, and then I put it this way. The outcome will always be the same, so don't be scared. Choosing the side where to drop which gametes, the outcome is the same. And now I'm going to cross here. This square here. This square here represents a cross, you should understand, a cross between these, so it's still a cross, and this. So I join them together and I'm going to put the outcome inside there. It would be capital D, because I'll always start with the dominant one, and lowercase d. So that's what I'm going to write in there. And this box here also represents a cross between this one and this one. So I'm going to do again the same. Capital D and lowercase d. And here is going to be a cross. This box is between this one and this one. I'm going to put the outcome. Lowercase d, and this box is between this one and this one. So I'm going to put the outcome here, lowercase d. So you see the way I'm crossing them, it's in this manner here. Take from here and then across here. I take here and then I cross with this. Take here and then I cross here. Take here and then I cross with this one. So that's the way I'm doing. It gives me the exact same outcome like I would have gotten with the schematic representation. Now, what process is taking place here? The process that is taking place here at this point is fertilization. So we're going to write here fertilization. And please don't write fusion. Fusion is not correct. Although some of our textbooks have it as fusion, but we need to learn how to correct some of these things ourselves. Fusion is not fertilization, but fertilization is the fusion of the sperm cell nucleus with the ovum nucleus. So when there is fertilization, when we're fusing these two nuclei here, there must be procreation. Something must be born out of it. Then we can talk about it as fertilization. Nevertheless, I can take two objects, let's say they're plastic, made of plastic, I melt them and then I fuse them together, but there will be nothing born out of it. So that's what we should know. Fusion is not fertilization. We're going to cancel that concept. It's not correct. Even if somehow we're trying to accept it because the mistake has been made already and many people are using it, but at our level, we should correct it. It's not correct. This is fertilization. Now, next, we ask ourselves, what do these actually represent? These that you have here represent the possible genotypes of offspring. In this case here, we're talking about the first generation offspring. I'm saying the possible. Why? Because it is not stated that if these two parents give birth to 4 children, each will be capital D's, lowercase d's, 2 lowercase d's all the time like that. No. It's possible that all of their children, even if they give birth to 16 children, it's possible that all of them end up with this kind of genotype of lowercase d, lowercase d. It's very possible. So our Punnett square here, or our genetic cross here, simply represents all the possible options of the outcomes in the F1 phenotypes or genotypes of the cross between this male guinea pig and this female guinea pig with their respective genotypes. Whatever number of children they can have, the children will only have genotypes that are part of the results that we see here, nothing extra. So for example, their children would never have a genotype capital D, for example. It cannot be possible. So the only possibilities are what we have here. So nevertheless, this line here shows genotypes, the genotypes of the offspring. And because we've seen their genotypes first for any other thing, because it's the genotype that we see first here in this line, we are going to therefore start our next line with the F1, since they are the first filial generation here. We're going to start with F1 genotype. That will be our F1 genotype. We're not starting here with the phenotype, mind you. And then in this F1 genotype, we ask ourselves, how many of this type do I have? I have 2. And how many of this type do I have? I also have 2. So this is to avoid writing capital D, lowercase d, uppercase d, lowercase d, lowercase d, lowercase d's, as if uppercase d and uppercase, lowercase d and uppercase d and lowercase d are different from one another. No, they are not different. So what we are doing here, we are putting like terms together. How many of those do I have? I have 2. I'm going to do that. And 2 of these ones as well. That is the genotype. That's how I write out what I have in that table. Now, next, I'm going to therefore go with the phenotype here. Phenotype. I'm going to have, I'm interpreting from the results I have here. So I'm going to have, quantify it as well, always quantify 2 dark fur, 2 white fur. Because yes, of course, this here gives me dark, and this here gives me white. So that's what I have. Now, with that, I think our genetic cross is done. But then let me remind here on the phenotype. This is also where most times, because people have seen this here, they'll come back here and say 2 heterozygous dark fur. No, like I said, those terms, heterozygous, homozygous, are for the genotype. Or they'll come here and say 2 homozygous white fur. They are for the genotype, not the phenotype. Let us remember that. Now, the most important thing that we need to know here, our question, probably I've forgotten to say, is out of 6. It's a typical 6-mark question. The most important thing that we need to note here is we ask ourselves, how are our scripts being marked? This is very important so that whenever we do our genetic crosses, we know exactly already where the marks will be allocated, so that we also avoid losing marks unnecessarily. So we're going to do a rundown to see how it's being marked, so that next time when you do your next genetic cross, you're going to know exactly what not to leave out, especially out of laziness. Because when you say genetic cross, people dive straight to a Punnett square, leaving out the most essential. So when we're looking at your paper, this is what we're doing. Ask ourselves, did this person write P1 and F1 in case it was about the P1 and the F1? If they said second filial generation, then it's supposed to be P2 and F2. So we ask ourselves, did this person write P1 and F1? If it was written correctly, we give you 1 mark. So if that was written correctly, you get 1 mark there. Next. We ask ourselves, did this person write meiosis and fertilization? If it was written, then we give you 1 mark as well for that. So you should realize, if, for example, you wrote P1 and then you forgot to write F1 or vice versa, you lose that mark. If you wrote meiosis and you forgot to write fertilization and vice versa, or you wrote fertilization, forgot meiosis, in the one that you forget, you lose 1 mark. Next, that's already 2 marks out of 6. We look at the phenotype. If well stated, not just dark versus white, then we give you 1 mark, provided, very important, we have this X here. This X here means mating. It means copulating, it means sex. So if it's not written there, then you lose that mark. Same with the genotype. If the genotype is correctly written here also, provided there's an X here, then we give you 1 mark for that. And for the gametes, sometimes optional, there must also be an X here. Sometimes it's optional as well. So, or it is best to always have that X there for the gametes as well. So put it in your mind that it should be a must, because the gametes have to cross with each other, they have to fuse, they have to fertilize one another. So the gametes, correct gametes written, I can decide to credit the mark here, but if you've drawn a Punnett square, I can credit the mark for the gametes down here on the Punnett square. So 1 mark for correct gametes and another mark for correct genotypes of the F1. And if you count the marks now, already I'm with 6 marks already. That is 1, 2, 3, 4, 5, and 6 marks already. That's already 6 out of 6. Now, I'll explain. The genotype here, I already gave that mark here, but it's always essential to write out your genotype, always, so that you can clearly see from there how many are expressing a particular phenotype, so you quantify everything. Also, it will help you in case they ask you about ratios or percentages or proportions, then you can easily work it out because you've quantified everything. Next, I'm going to have the phenotype here. If the phenotype was given correctly, then I give you 1 mark for that as well. So we are ending up here with a total of 7 on 6. Should not be weird to you because the extra mark that we have here could be a compulsory mark. It means until you have that phenotype correct, that's when you're going to get 6 on 6. Meaning, if you failed this one here and got all the other 6 marks, you end up with 5. And if it's not made a compulsory mark, at least you have 1 extra mark saying that if you failed any of these other points, at least you'll still end up with 6 on 6. So in general, guys, phenotype opens the party, phenotype closes the party. Thank you.